A-level biology, topic by topic. A little bit later than we advertised, but still, we'll have a go. I want to look at some carbohydrates today. Carbohydrates, one of the main food and energy sources for the body, and glucose is not normally available as glucose, but as a whole variety of different molecules joined together, large polymer molecules that we break up. So I want to start off having a look at glucose, what it is, and what we can do with it. So let's dive in and have a look at this. Right, now what I want to do is, let's zoom in a little bit, have a look at glucose, and I'm going to draw it first and then I'm going to try and make it for you with some models. So we've got this basic 6 ring. I'm going to try and draw it in this sort of 3D way. It's not terribly even, and what we've got is an H above here and an OH that way, and then we're going to have it reversed this way, and then we'll have it reversed this way. Up here we're going to put a CH2 OH group, which is the same, and here we're going to have an H and an OH. So there is our model of what we call alpha glucose. There's another molecule which is very, very similar. I'll see if I can perhaps draw this one slightly better. And the answer is no. It's exactly the same as before. We've got the CH2, OH, we've got our H above, OH below, OH above, H below, H above, OH below, but here we're going to have the H and the OH round the other way, and this is beta glucose. So we've got 2 different molecules of these hexose monosaccharides. Now when we look at these, the carbons are numbered in a clockwise fashion, beginning with the carbon to the right of the oxygen. 1, 2, 3, 4, 5, 6. So we've got 2 structural variations here, alpha and beta glucose, and simply the only difference here, the hydroxyl group on carbon 1 is on the opposite positions. So let's see if we can make this molecule, and we're going to make it using some statins. So let's have a look at the statins. They make nice space filling models of this, so we'll use this one to try and show what we've got. So first of all, we've got a 6 carbon chain, and one of these we've got a oxygen atom here replacing. So there is my basic ring. Now let's start populating this with what we need. So we'll do the awkward one at the top, and that's going to have here an OH, on there. There we go. And then if we start with this one, we've got two choices where we can put our hydrogen and our OH on. So I can put it this way round, or I could put it that way round. Let's see how we go on with this. So I've got to try and put it down a little bit so I can actually make up what I'm going to do. We're going to get some OH groups put on the system, and we'll see where we go from. Right, here we go. So if we take making our alpha glucose, then let's just shove on to there a hydrogen, and I want to try and put here the groups, so we've got them that way round. So this one's going to go up, and that one's going to go down, and then this one we're going to have down, and this one up, and that's going to leave me with this one, which is the alpha glucose form, which is this one's up and that one's down. And you can see that this makes quite a messy sort of molecule. It's hard to see what's going on. I've got a mixture here of, if you're interested, new statins and old statins, and the advantage of having this is the new statins are much stronger. Right, so here is our molecule. Let's look at this with some Molymod and see if we can see it looking slightly differently when it's not a space filling model. So have a look at what we've got. We've got a couple here of glucose molecules. Let's zoom out so you can see them quite well. Right, now starting at the top end here, we've got this carbon COH, and we've got going up, so that one's, if you like, here we can think of this one as there's the up, there's the down, here's the up, there's the down, here's the up, there's the down, and in this one we've got the OH up, so this makes this beta glucose. Let's compare this with another molecule, and I've got a different molecule here, so I'm just going to make this into glucose. This happens to be a fructose molecule. Now I'll just check that this is all okay. If I can get my hydrogen on anyway. Perhaps not. Right, what I'm going to do very simply is I'm going to get that fixed, and off scenes we're getting that one sorted out. Right, let's have a look and see what we can do with this. So all I've got with this molecule, you can't rotate this molecule around to turn it to the other way. It's sort of stuck in this form. Right, let's have a look and see what we've got with this one. So let's start with it round the same way. We're not right here. Right, I think that looks about right. So, carbon 6. This one we've got a choice of up and down, and it's exactly the same. This one we've got this one up, down. Here the hydrogen's up, that's down. And this one's in the same orientation. So this one will be the other way up, and there we have the difference between alpha glucose and beta glucose, and there's not a lot in it. So let's see what happens if I change both of these to alpha glucose. So I'm going to change my beta glucose. Slightly off camera there, where I just try and push the bond in. There we are. So I've got two alpha glucoses. Now, when we get alpha glucoses reacting together, we form what we call a glycosidic bond. Let's draw this first to see what happens. So I'm going to take now two alpha glucoses. And I'm not going to try and draw these as well as I did before. And with these I've got my CH2OH at the top. And if we put in all the bonds that we have, I've got the OH's at either end at the bottom. This one's going to go up, this one's going to go down, so we've got alpha glucose here. We're going to draw this one in the same OH, just shoving them round in the same pattern. Now what we're going to do is we're going to take these two OHs, and we're going to undergo a condensation reaction. So if we draw out the same structure as before, what we're going to end up with is, with this here, I'm going to link these and I'll leave this one here. For the exams, do you need to be able to draw these things like this? And the answer is, of course, yes. So here we've got our structure of a glycosidic bond, and this is a 1,4 glycosidic bond. Because this is, if we label our carbons, carbon 1, 2, 3, 4, 5 and 6, so we're looking at 1 joining with 4. So we've got this name, the 1,4 glycosidic bond, and because it's a condensation reaction, we also get here plus water. And if I want to name this I would name as a disaccharide, and in fact it's 2 glucoses joined together, and I'm going to call that maltose. Let's see if I can join the model together to do this. So we've got my carbons, my organised here with my 1,4, and I'm going to organise my other one in the same way. It's set at being 1,4, and what's going to happen is water is going to be eliminated from here, and the 2 molecules will be joined together, and there I've got my water molecule made and it's eliminated. So here is my disaccharide, that of maltose. Now I didn't do that on the space filling model because really it's very difficult to try and see what we've got. We have quite a lot of these disaccharides. We've got a whole variety of them. So we've got 2 glucoses that are called maltose, but we've got some other ones that are ones that you ought to be familiar with. The next one is where we have 1 glucose and 1 fructose joined together, and this is sucrose, which everyone knows as common sugar. We can also manage to take 1 glucose, but this time join it with 1 galactose, and this makes another sugar which is called lactose. Fructose here is fruit sugar, much sweeter than glucose, and when I get students to try galactose, they usually complain it tastes more like sort of more flour than anything else. They find it's not really a very good sugar. There are some other sugars that you ought to be aware of, which are monosaccharides. And these are probably ones that should be familiar to you, and these might be a sugar called ribose and its friend deoxyribose. And these, of course, are found in things like RNA and DNA. Many of these alpha glucose molecules can be joined together by their glycosidic bonds. So we're not talking about one little bond here, but we're talking about taking a whole string of these different ones and basically joining them together to make really quite long chains. And I'm making this bond there a little bit bigger because I'm trying to highlight a point. So we've got this chain which is basically a little bit bent here, and this glucose, which is made all by the photosynthesis in plant cells, is going to be stored not as glucose, but it's going to be stored as normally a chemical called amylose. So this is just a little bit of a molecule of amylose, and amylose here is formed simply by these 1,4 glycosidic bonds. The angle of the bond means that this is a long chain which is very, very slightly twisted. You can see it's very, very slightly twisted, and this means that when amylose is formed, it actually twists to make a helix. So what we're going to get here, perhaps a little bit keen there, is something that's going to look like this. It's going to have all these sort of twists in it, making a nice helix, and this helix will also be stabilized by lots of hydrogen bonding between some of these water molecules here and the hydrogens there on different atoms. Now this is going to make this, which is now a collection of saccharides, so I'm going to call this a polysaccharide, and it's going to make this polysaccharide more compact, less soluble than the simple units that made it up, and this is going to form something which we'll be more familiar with by the name of starch. Now we can get another type of bond occurring, and let's just take these apart. And I'll zoom out for you. Right, so we're back with our molecules ready to start, and what we can, instead of doing, we can join these in a different way. And the way we're going to try and join these is not with a simple bond, which is a 1,4 glycosidic bond, but sometimes we can get something that's going to go with a 1,6 glycosidic bond. And in this case, then we're going to remove a water from here. And now we're going to get this type of structure. So this is going to give me a 1,6 glycosidic bond. We better have a look at this and draw it out. So let's take my two glucoses and instead of this time, so I'm just going to put down, down, up, down, and this CH2OH. Now we're going to bring this and I've just turned it round very slightly, that molecule. Here we're going to lose with a condensation reaction here. So this time we're going to get a 1,6 bond. And we'll just put in all the other necessary bits and pieces, and there, this is a 1,6 glycosidic bond. Now let's see what that causes with problems. So if I've got an amylose chain and then somewhere in there I might have a 1,6 bond, and on there I could have another 1,6 bond, and somewhere along here I can have another 1,6 bond. We still have our 1,4 bonds working, so I could quite simply on here, let's just quickly and very easily draw out my thing that we've got. We've got that going down there, going on to my CH2, and here we will have our 1,4 glycosidic bond, and going along here we might have our 1,4 glycosidic bond, and along here we might have our 1,4 glycosidic bonds. So in addition to our 1,4s, we've got this extra link in here. Now, this starch is called Amylopectin. So amylose was simply made by 1,4. Amylopectin is made up from 1,4 glycosidic bonds between the alpha glucoses, but also some other 1,6 glycosidic bonds. And this occurs about every 25 glucoses. It's a very approximate number, but it can't sort of get these chains to fit in if they're any closer. It becomes way too unwieldy. In the way this works, if I want to try and break down a chain, let's look at the difference between amylose, and I want to look at a chain of amylopectin. If I'm a molecule that's going to try and devour this, I'm just going to latch on at the end. This is an enzyme coming along, and it's going to chop the glucoses off one by one as it moves down the chain. It cannot start in the middle, has to start on the end. So if I want to break down amylose, this molecule has to start, this enzyme has to start at one end and work its way through. More efficient if I could start on several places, and this is what's done in amylopectin. In animals, this, and fungi, this is stored, and this is glycogen, whereas this one tends to be starch. So this speeds up the process of trying to break it down. So it's still here branch, so it's also going to be insoluble, and it's also going to be quite compact. So we have here a much faster means of getting glucose using glycogen, the amylopectin layout, rather than just using amylose starch. Let's look at a different molecule, and this molecule I want to look at is one that should be fairly familiar to you, and this is cellulose. Cellulose is made not up from alpha, but made up from beta glucose. So let's take my amylopectin, and what I'm going to do with this, zoom out, is I'm just going to turn these both into the right molecule. So let's get the orientation right to start with. These now both are alpha glucoses, and I've got them there in the same format. And if I lay them down the same way, you can see we've got both alphas. Now I'm going to turn this into a beta glucose, and I'm going to do that by taking this one off. that's alpha form. This is my beta form. So just this quick swap round, and now we've got these molecules of beta glucose. Let's see what happens when we try and join these together. When these are going to go, they're going to work in exactly the same way. We're just going to get a molecule breaking up here, and we've got a slightly different structure going to happen. Let's draw it out first. It might be a bit easier to see what's going to go on. So let's take my two molecules of beta glucose. In this one, because it's beta glucose, we're going to have the OH in the up direction, and this is going to react with another glucose, also a beta glucose. So this one here is in the down direction, this one's up, this one's down. When this is going to join, what we need to do is we need to flip one of these molecules. So that's going to join there. So let's try and sort this one out, because these are not the same way round. This is the CH2OH up here. I better put that one in to make it clearer. So let's make this. I'm going to change the orientation here slightly. So let's do that one up. That's going to be easier to see. I'll do this one the same way up. So up, down, up, down. Let's put this CH2OH in there. I'm going to flip this molecule upside down, so this time we're going to see it drawn this way up. So I'm going to have my CH2OH this way, and that's going to mean that this one's going to be that way round, that one's going to be that way round, that one's going to be that way round, that one's going to be that way round. So now these are in the right orientation to work, and if I draw this out much more simply, what we've now got is this one with its up bond corresponding to this one with its down bond, and on this end we're going to have the down bonding with the up on the next one. Now what this gives us is, with this up down type of configuration, is a pretty straight molecule, and this is going to be a lot straighter than the other one, the amylose. So the amylose made a spiral, whereas this is unable to coil. And it's also unable to form branches. And this gives cellulose a lot of its properties, because it's going to mean that our chains here of cellulose are going to have bonds going across from one chain to the other. And once we've got these long chains, which we're going to call now a microfibril, will be joined together. Lots of these microfibril chains are going to be joined together, and they are going to make a macrofibril, and lots of macrofibrils are going to get together, and they are going to form here a cellulose fibre. And this stuff, thank goodness, is insoluble. It needs to be insoluble because this is the rigid stuff in a plant that holds the plant basically together. It's starch in this form, cellulose, same glucose joined together, which is now insoluble, that forms the material. It forms all the cell walls of the plants, a rigid sort of scaffolding like structure, and this makes cellulose such a hard, durable substance. And you can shove this in the washing machine on hot and it survives remarkably well. So I want to finish by looking at how we could actually do some testing for this. Let's have a look. Trying to test for some of these molecules, we're going to use a calorimeter. So here is my calorimeter, and I've got here a collection of cuvettes. Now my cuvettes here are round rather than some of the more familiar sort of square ones, and my calorimeter here, we plug this in to the computer. I can open up my hole, I can put my sample in, and we'll shine a light through this substance, and we can measure the amount of absorption, or you can measure the amount that is sort of transmitted through. So here, my little calorimeter. Now I want to explain how we can actually use experiments to make this work. And I'm not going to do this one because it takes way too long. But I'll tell you how it works. I've got my colorimeter, and we're going to try and use an experiment to try and detect my sugars. So you ought to know a test for sugar. So our test for a sugar is Benedict's. And when it's a sugar here, I actually mean it's got to be a reducing sugar. Benedict's test, then, what we do is we take our test tube, we put some sugar in, and we put some Benedict's. And hopefully you will mix the two. We then place this in a hot water bath, probably for about 5 minutes, and we get different colour changes. So Benedict's is blue, so if we've got blue and it remains blue, we've got no reducing sugar. If it goes a sort of a green colour, this is very low amount of sugar. If we get more of a yellow colour, we've got a low amount of sugar. If we can get some sort of brown colour, then we've got a medium amount of sugar. And if we can go more sort of brick red, then we've got a high amount of sugar. And this is very much a sort of qualitative test. Now, how can we make it a quantitative test? Well, here we're going to use the colorimeter. To use this, what I've got is a sample of sugar that I know nothing about. So I've got a sample of my sugar, and I've just done this Benedict's test on it, and it's one of those colours up here. Not any help. So what we do is we do a whole series of sugar tests. So I make up a 0.1, 0.2, 0.3, 0.4, 0.5 molar amount of glucose solution, and to all of these we do the Benedict's test. Now once we've done the Benedict's test here, and I don't know what the values of those are really going to be because it depends so much on this sugar, what we can then do is we can then plot a graph of the absorption PTION, and I got the concentration of my sugar here, and if it's going to be absorption, it's going to be a graph that's probably going to look more like this. And having got this graph, we then test this sugar sample and we find out where on the line it's going to be, and we can then read off on this graph what the concentration of this sugar really is. So that is all about carbohydrates, having a look at the different structures, the alpha, the beta structures of glucose, how they join, the alpha making up either just straight chains, which is amylose, or the branch chains with the 1,6 glycosidic bonds instead of just the 1,4, so it's got the 1,6 and the 1,4 glycosidic bonds, and that's making amylopectin. And if we have beta glucoses, it will only form 1,4, but the molecules are up, down, up, down, up, down, and this makes a very straight molecule, which is insoluble, which is great for making fibers such as cotton. If you like this, please then subscribe, and I hope to see you next time when we'll look at some more A-level chemistry topic by topic. I'll see you then.